Hello, it's Miss Bell again. So this PowerPoint is on cellular transport. So we're going to be looking at how cells transport things in and out of the cell. So at this time, I want to take a moment to remind you that you do need to be following along in your workbook and following the agenda each week. Because the workbook has some very important reading on this stuff. So when you do your workbook sections, you should be reading it and following along with it, and I hope you're doing that. And you'll notice that it coincides directly with the PowerPoints. So this PowerPoint goes along with section 3 in your workbook. By the end of this semester, we're going to go through the entire workbook. So we're on section 3 now. So anyway, just a review of the cell membrane or the plasma membrane. Remember, it's made up of primarily the two P's, the phospholipid bilayer. Let me change colors. Goodness gracious. The phospholipid bilayer, which is the two layers of phospholipids, and then the proteins. I'm sorry if you hear my toddler in the background. This is my only chance to get this done. So anyway, the two P's, phospholipid bilayer and proteins that are interspersed, and there's also some cholesterol in there. And if you recall, cholesterol is a type of lipid. So anyway, the cell membrane is considered amphipathic, which means that it has both a hydrophobic and hydrophilic region. And so that's talking about remember your phospholipids. So that's what the bilayers are. And so your hydrophobic is going to be your tails. And then your hydrophilic is going to be the phospholipid head. And that's because the head is polar and the tail is fat, the lipid portion of the phospholipid, so it is nonpolar. So remember, polar is having a partial positive and partial negative end on different ends. So if you recall, water is polar. So remember, Mickey Mouse's face is negative and Mickey Mouse's ears are positive because of the way the electrons are arranged. Lipids do not have a positive end and a negative end like water does, so that makes them nonpolar. So here is a great picture of the cell membrane. So I want to point out a few things. So you have your two layers. So here's one layer, and here's your other layer of phospholipids. So basically, with the phospholipid bilayer, the inside is going to be hydrophobic, and the orange outsides is going to be philic. Okay, so the outside of the cell is going to contain water, so it's going to be hydrophilic. The inside of the cell is going to contain water, so it's going to be hydrophilic, if that makes sense. And then you have various proteins within the cell membrane, and then here's showing you a carbohydrate chain that may also be on the cell membrane. And we mentioned several of those things in the previous PowerPoint. Okay, so if this just kind of goes into the hydrophobic, hydrophilic thing again, just to refresh you on it, because it is very important for this unit. So if the bilayers, the two layers of the cell membrane, were looked at individually, then you would notice that each layer has a hydrophilic portion and a hydrophobic portion. So the hydrophilic portion is that phosphate head. So if you recall, it's the phosphate group attached to the glycerol, and that forms the inside and the outside. And so recalling, hydrophilic is water-loving, and so that means it likes water. It's similar to water because it's polar. It's attracted to water, and it can also be dissolved in water. Then you have the inside portion of the cell membrane, and that's the hydrophobic tails. So that is your two fatty acid tails, and if you recall, the cell membrane sits tail to tail. Okay, so anyway, this is nonpolar, so that means it fears water, it does not like water, it repels water, and it does not dissolve in water. So again, inside the cell membrane, you have some proteins, and there's different types of proteins. And we talked, a lot about those in the previous PowerPoint. So remember, an integral protein is going to go all the way through. It's going to penetrate the entire membrane. So it's kind of like a pipeline. So it goes straight through, inside to outside. And it's going to help you transport things in and out of your cells. Peripheral, okay, your peripheral vision is like your outside vision. So they're going to be either attached to the inside or attached to the outside of the membrane, and they're not going to actually go through the membrane like an integral protein would. These are typically either going to be like enzyme proteins, or they might help with form the structure of the cell membrane. Then you have ion channels. What do you think comes through an ion channel? Probably an ion. Okay, so ions are charged particles, if you remember. They're either positive or negative. So because of that, they're not going to go through that nonpolar fatty acid tails part of the bilayer. So they are a protein. They're kind of like a pore that the ions can just pass through. And there's certain ions that your cells need, and they need it in certain amounts. And so those ion channels are going to allow the passage of those ions. So you'll have like a positive ion channel and then a negative ion channel, depending on the charge of the particle that needs to pass through. So, structurally in the cell membrane, both protein and cholesterol are going to contribute to the structural portion of the cell membrane and the stability of it. Okay, so they help form the structure of the cell membrane. And again, the proteins can form different channels, pores, or even like a gate-like passageway to allow substances that could not go through the nonpolar interior of the bilayer. And you do have some carbohydrates in the cell membrane. So with the carbohydrates, what they do is they look at cell-to-cell recognition. Okay, so what that means is they can tell if it's a self versus non-self cell. So basically they can tell, does that cell belong in my body, kind of thing. And so those carbohydrates are going to hopefully not allow different viruses, bacteria, and other bad things to enter your cells. But again, we're human. Everything doesn't always work perfectly, and some pathogens are very complex. And they can get through that carbohydrate kind of like a gatekeeper. Okay, what kind of organic compound makes up the double-layered membrane of all cells? Hopefully you were thinking of the phospholipid bilayer, so that would be lipid. Okay, so let me move this a little bit. There. So the cell membrane, again, is selectively permeable, and this should be a refresher based on the previous PowerPoint. So what that means is it selects what enters and leaves the cell. So it controls what enters and leaves the cell. So it's like a gatekeeper. And how it does that is it senses what the environment is like outside the cell versus inside of the cell, and it kind of balances things out. And how it balances things, well, the cell's very smart. It knows what it needs. It knows the perfect amount of water and salt and calcium and all these different atoms. It knows the perfect amounts that it needs to function. And so if it needs something, it brings it in. If it needs to get rid of something, it gets rid of it. So the cell membrane is selectively permeable because it's only going to allow what it needs and what needs to leave in and out. So it controls what enters and leaves the cell. It's kind of like my door on my classroom. So I have a lock on my door. We're supposed to usually keep our classrooms locked. And so that way, if we need to keep people out, So we need to keep people out of our classroom or out of your house, for example. You might keep your doors locked at your home. So if you keep your doors locked at your home, only the people that have a key are going to be allowed in. So you're selecting who enters and leaves that house. So anyway, another thing the cell membrane does is it keeps the cell separate from the rest of the environment. So it helps it remain separate. So Singer and Nicholson, they developed the fluid mosaic model back in 1972, so not that long ago, considering. So they developed the fluid mosaic model. And it basically is referring to the structure of the cell membrane. It's saying that, so if you think about this is the basic structure of the cell membrane. It's saying that the cell membrane has a bilayer, a phospholipid bilayer with proteins embedded. So that's kind of what we've been talking about. So Singer and Nicholson, 1972. So again, this is another picture of the cell membrane. So we have some proteins here. So if you notice, this is a protein and it goes all the way through. Okay, it goes all the way through. And then notice this one does not go all the way through. So this protein, this one here, actually is probably an enzyme. So anyway, you got some carbohydrate chains going through here, some proteins, of course your phospholipid bilayer. Okay, so the plasma membrane of a cell is selectively permeable, which means it, hopefully you picked, allows some materials to pass because it selects what can pass. Okay, so the cell membrane and temperature. So one kind of unique thing that happens with the cell membrane is based on the temperature, the membrane fluidity changes. So just real quickly, I want to mention to you, we talked about the fluid mosaic model. One reason they called it fluid is because things can fluidly pass in and out of the cell membrane. And the mosaic comes because the picture you previously saw they felt like it tended to look like a mosaic tile. Those are the little tiles that make the pictures. So anyway, so this gets into the fluidity and the solidness of the membrane. So as the temperature decreases, so as it gets colder, the membrane remains fluid. And what happens is those phospholipids get closer and closer and closer together. And then eventually, as the temperature gets colder and colder, the membrane will solidify. Okay, and this should kind of make sense because things tend to, especially things that contain a lot of water, are going to, as it gets colder, they're going to become more solid. And so that should make sense to you. Anyway, so the types of lipids that the membrane is made of helps determine what temperature it will solidify at. So again, when you're talking about just anything, water versus salt water, for example, or water versus a popsicle, they're going to solidify at slightly different temperatures because the what's inside of them is a little bit different. So if you have a lot of phospholipids with unsaturated hydrocarbon tails or unsaturated fat, then it will remain fluid at a lower temperature. If you remember, I showed you some phospholipids that look like this and phospholipids that look like this. Okay, and the ones with the kink in the tail are unsaturated. So what that means is when I put these, they can't get as close together. Sorry, not a great drawer. They can't get as close together as, say, these can. Okay, so this would be unsaturated, and this would be saturated. So you can see saturated can get closer together. So anyway, that kink, right here, that kink is where that double carbon-to-carbon bond would be located. So anyway, that kink makes it not be as closely together. So it's going to be harder for it to solidify, so it can be colder and colder. So it's going to remain fluid and not solidify until it gets to a much lower temperature comparatively. So, the steroid cholesterol, I mentioned earlier that it can be found in the cell membrane. So, it just kind of is between the phospholipid molecules, and cholesterol is going to be in animal cells. Anyway, warmer temperatures. So this is different because warmer temperatures, cholesterol becomes less fluid, and it restrains the phospholipid movement. So anyway, so it lowers the temperature required for the membrane to solidify because it too does not allow the phospholipids to be closely packed. So basically, it's creating an extra space. So this is why cholesterol is called a temperature buffer. So it causes the cell membrane to resist changes in the fluidity, and that fluidity and temperature have a direct relationship. Okay, so we talked about homeostasis before, and homeostasis is that stable internal environment. You need just the right amount of water. Doesn't matter if I live in water, I still need to have the right amount inside my cells so that my cells can work properly. I need the right amount of sugar. I need the right amount of oxygen. All those things that you know that cells need. And the cell membrane is going to help the cell maintain that homeostasis because it controls what enters and leaves the cell. So that's how it controls that's how it maintains homeostasis, is by controlling what can enter and leave the cell. So there's 3 steps to maintaining homeostasis and the response for homeostasis. Um, there we go. So the first step is going to be the sensor. So this is when your body notices that something is out of balance. Maybe you have a fever. Maybe you're dehydrated. But whatever the fact may be, this is when your body first senses that something is not as it should be and not within homeostasis. Then you have your control center. That would be like your brain, and it receives a message from the sensor saying something is out of balance. You need to fix it. And then you have the effector. The effector is kind of the last part. It receives the message from the brain for you to fix whatever the problem is. So, maybe the sensor is your cells notice that there's too much water in them. And then so that goes to the control center to your brain, and then your brain sends the message to the effector, maybe your kidneys, saying, okay, you need to urinate more to get some of this extra water out. So, homeostasis is very important to keep the cell alive and well, and metabolism is a major thing that must be maintained through homeostasis. So this is basically going to be involving your energy conversions in the cell. So this is basically creating ATP and releasing the energy in it. So storing and absorbing the energy in ATP. So you have catabolic and anabolic reactions. If you recall, we talked about these in the biomolecules unit. So catabolic are going to be breaking down reactions, and anabolic are going to be building reactions. Okay, so in your body, cellular respiration breaks down glucose, sugar, into carbon dioxide, water, and energy, whereas photosynthesis would be an anabolic reaction. In plants, photosynthesis makes sugars from carbon dioxide, water, and sunlight. So that brings us to cellular transport. So, if you think about the properties of the cell membrane that we just talked about, you'll probably recall, and it probably will make sense to you, that nonpolar molecules and small molecules are able to easier pass through the cell membrane. Now, water is going to pass through pretty easy because your cells have special things to help that pass through. Carbon dioxide, oxygen, these are things that you know that you need in large amounts, and so they're going to pass through pretty easily. Then you have your large molecules, like glucose is a larger molecule, amino acids, things like ions that have a charge, proteins, most polar molecules are not going to go through. So anyway, these things are going to be harder to pass through, so they have to go through usually a protein or possibly an active transport method. So again, ions and most polar molecules cannot go through the cell membrane because they can't get past that nonpolar area. They can't get past that nonpolar area. So here is a picture of the cell membrane. So this is a great picture because it shows you the outside of the cell versus the inside of the cell. And it shows you your lipid bilayer. Here, the yellow, here's you see your cholesterol right there. And if you notice, the cholesterol doesn't allow the phospholipids to get so close together. Here it shows you some carbohydrate chains. Notice they're kind of on the outside of the cell, so they can determine, oh, hey, is this cell that's coming up to the outside, is that going to be a cell like me or something bad? Notice the channel protein. It's like a pipeline because it stays open. And this is a gated channel protein, so if you notice it's kind of closed here. This is a transport protein, so we're going to talk about how that works. Notice one side is open here. It's actually going to change the shape to allow, to accommodate whatever the molecule is that's going through. And then this is a receptor protein. And then this is just showing how the cytoskeleton is going to connect to, you see it kind of comes up and it's connecting to the cell membrane. Okay, so there's two ways or two types of cellular transport, passive and active. So if you think about like the basic word active versus passive, they're opposites. So if you learn, this is a good unit to learn like if it's opposite or learn through exclusion. So passive transport is kind of going to be just what it sounds like. If you're active, you're using energy. So if you're passive, you do not use energy. Okay, so it does not use ATP. So if you see ATP or energy in any picture, it is not passive, it is active. Active uses ATP and energy. Now, for it to be passive, it needs to go with the flow or go with the concentration gradient or go down the gradient. Now, we need to mention what a concentration gradient is. So concentration gradient, so first off, concentration is the amount. Okay, so concentration means the amount as far as science is concerned. So if we say it goes with the concentration gradient, then the concentration gradient you need to write this. Concentration gradient is going to be your difference in amounts. Okay, so this is having a high amount and a low amount. If you have equal amounts, you do not have a concentration gradient. Okay, so if you look at this picture here, I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. We just won't count this one because it's on the line. So you have 12 particles on this side and 3 blue particles on that side. So here you have the high amount and the low amount. So that is a concentration gradient because there's a difference. If there were 6 and 6, then it's in equilibrium. There's no concentration gradient. So you have to have a concentration gradient for passive transport to occur, and it's going to go with the concentration gradient. So it's going to kind of go with the flow. So if you look at this picture and you think about like a room, if you have like a classroom full of like 50 people, that's going to be pretty packed. So is it going to be easier for you to go from the high amount in the classroom to no one in the hallway? Is it going to be easier to get out or to pack in, to enter a room that's already slam full? So typically it would be easier to exit a very crowded room like that. So you would be kind of going with the flow. So the direction that passive transport's flow is from high amount to low amount. So you must pay attention if you're given a picture the direction of the arrows, because all I have to do is flip this arrow the other way and it will be active transport. So passive transport goes from high to low amount. It uses no energy and it goes with the concentration gradient. Now, all three types have all these three characteristics in common. It's very important that you understand these things. When we get to active, you will notice they are opposites. The characteristics are opposite. So if you can just remember one, then you can know the other is opposite. So again, in all three, the direction is going to go from high amount to low amount. So this is supposed to be like an animation. It's supposed to show you that there is, the beaker is empty, and then you start out with ink at the top. So you would have your high amount here, and then your low amount down here. I mean, you can kind of tell here will be my low amount. And the ink travels from high to low until the entire beaker is the same color, yellow. This would be like dye or ink or food coloring in a beaker, and how it would move randomly throughout the beaker to eventually become equal. So again, I can look at this picture and I can see that, let me change colors. I can see this is my high amount and that this is my low amount, and it's traveling from high to low. Notice it's traveling through the phospholipid portion of the membrane, not the protein portion. That will come in handy in a little bit. So again, inside of the cell, outside of the cell. This is just another picture up here going from high amount to low amount. So high to low, passive transport. So there's three types: diffusion, facilitated diffusion, and osmosis. Guess what all three types have in common? They're all types of diffusion. So all three are types of diffusion. If it is not diffusion, it is not passive. Okay? Because osmosis is diffusion of water. So you have regular diffusion, facilitated diffusion, and the diffusion of water, which is osmosis. So if it's diffusion, it's passive. If it's not diffusion, it's active. So diffusion, that's the most basic type of passive transport. If you have a concentration gradient, and remember that's a high amount and a low amount, if you have a high and low in that solution, then it's going to move from high to low. Okay, and this is a random movement of particles. So if you look, this picture, here's my high amount, here's my low amount. Notice the direction the arrow is traveling, okay, from high to low. Again, this is not going to use energy, and it's going with the concentration gradient, because all passive transports go high to low with the concentration gradient using no energy. So diffusion depends on that concentration gradient. Remember, that is your difference in amounts across the space or across a solution. Equilibrium is when it's the same. So you either have one or two states. You either are in equilibrium or you have a concentration gradient, a high and a low amount. Okay, so it's one or the other. You don't have both. So if there's no concentration gradient, okay, so let's say you have 5 on one side and 5 on the other. That's equal. It's not going to go from high to low. It has to travel from high to low. So there's no high or low. Okay, if we have 7 and 4, okay, it's going to go from high to low. Okay, and another important thing is that in equilibrium, the particles aren't just still. They're just moving back and forth equally. If 2 moves to the left, then 2 will move to the right. So the diffusion of a substance into or out of a cell requires what? Hopefully you picked a concentration gradient. Okay, so now we have facilitated diffusion. So if you understand vocabulary, to facilitate is kind of like helping. Okay, if I facilitate you changing your tire, then I helped you change your tire. So with facilitated diffusion, it's still diffusion. It's still going from high to low. it's still not using energy, and it's still going with the concentration gradient. It is still a passive transport. Okay, but the difference is it has a helper. It has a facilitator, and that facilitator is a protein. Okay, so basically facilitated diffusion is diffusion through a protein. Okay, so with regular diffusion, regular diffusion goes through your phospholipid part of the cell membrane, and facilitated diffusion goes through the protein part. Osmosis is a type of facilitated diffusion, so it also moves through the protein part. Okay, so if you just look up here, you can see that diffusion goes to the phospholipid part, facilitated diffusion and osmosis go through a protein. So things like amino acids, glucose, and other things, they go through facilitated diffusion. And again, this is for particles that cannot go through the phospholipid bilayer. Maybe they're too large, or maybe they are polar. So here's a picture of facilitated diffusion. Notice the particle is moving through the protein part of the cell membrane. So if the arrow is going through the phospholipid part and it's going from my high amount, notice the high amount, 1, 2, let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 versus 1. So this is my high amount and this is my low amount. And notice the arrow going down. If it's going through the phospholipid part, this will be diffusion. If it's going through the protein, this will be facilitated diffusion because it's still going high to low, it's still passive transport. So here is another picture of facilitated diffusion going. There's my high amount, there's my low amount, arrows going from high to low, and it's moving through that protein. So again, facilitated diffusion. Now I want you to take a look at this picture because this is a protein that is changing shape to help get this substance into the cell. Okay, so it if you look, it changed its shape in order to allow this substance to come in the cell. Okay, so that will be a carrier protein. It's changing shape to carry the substance across the cell. So again, an ion channel, that's going to be like a either positive or negative or polar pore that allows ions to pass through. And it's a protein, so it doesn't have to go through the hydrophobic interior. It moves through the protein. Okay. Oops, sorry. So you really need to know the 6 types of protein functions, or basically 6 types of proteins that you could see and what they do. So the first type is transport proteins. Transport proteins may be called channel proteins or carrier proteins or just a transport protein. And of course, they're going to help transport things across the membrane. So I showed you a couple of pictures of channel proteins already. So the channel proteins are just providing a straight shot to let molecules come through. So it's just an wide open pore. It doesn't do anything special. It's kind of just like a tunnel, pretty much, that allows substances to go into or out of the cell. Then you have a carrier protein. Like I showed you before, you don't see pictures of it as often, and they change shape in order to fit that molecule to help it cross over the cell membrane. So it's going to bind to it, change shape, and then push it in or out of the cell. So that's kind of the thing you usually would see is change shape. Either way, both channel and carrier proteins are going to help transport things across the cell membrane. Then you have basically enzymes. Remember, enzymes are types of proteins. So these are little proteins that are sitting on the outside, usually, of the cell membrane, and they basically have just an active site that's exposed that can perform different functions. Then you have signal transduction proteins. Okay, so signal transduction proteins are going to be, they're basically going to be like where hormones bind to. If you remember, so it's a binding site for typically like chemical messengers like a hormone. So again, kind of how so this would be like the enzyme, and this would be like the substrate, how the active site's exposed and it fits just perfectly. It's kind of the same way with these signal transduction proteins, except instead of for a substrate, it's going to be for a chemical messenger like a hormone to attach to. And certain hormones only act on certain cells. So, anyway, it's basically a place for like a hormone to connect to. So if you recall when we talked about hormones before, we mentioned a couple of different examples. So I'm going to use one. Let's see, cortisol, that's a stress hormone. So cortisol is produced in the adrenal glands. They're little glands that sit on top of the kidneys. So it's produced there, it goes into the bloodstream, and then it goes to other parts of the body. Well, let's pretend cortisol goes to your heart. It's going to have a binding site on the heart cells to go to. And then once it binds, it's going to act on that cell. Okay, so the fourth type of protein is going to be cell-to-cell recognition. Okay, so we mentioned that carbohydrates that are in the cell membrane can do this, but there's also proteins that can do this too. Basically, it figures out what cells are that come up next to it. Okay, so again, it's going to identify self versus non-self cells. Okay, and sometimes a glycoprotein can act in a similar function and identify what cells are up next to it or approaching it. Then you have intercellular joining proteins. And so some neighboring cells need to interact. Some of them need to stick to each other or even communicate. Okay, so for example, nerve cells are going to need to communicate to one another. So these are proteins that kind of let cells stick together and interact. So like if I'm muscle cells, everybody needs to contract at the same time, all the muscle cells in that muscle. So they would need to be joined, the cells would need to be joined, and they would need to be interacting at the same time. Then you have proteins that attach the cytoskeleton to the matrix outside of the cell. So if you recall, I showed you a picture just a little while ago of the cytoskeleton, and the proteins were kind of into or merging with the cell membrane. Well, these proteins are also going to help connect the matrix outside of the cell with the cell. So that way your cells aren't just kind of all over the place. So your third example of passive transport is osmosis. And like I said before, osmosis is a facilitated diffusion. If you notice, the water is going to primarily travel through a protein. That particular protein is called an aquaporin or a water pore. So it's a water channel protein. It's a channel, so it's just wide open, and the water is going to easily flow through it. Now, you'll notice that there are some water molecules in the phospholipid bilayer. And notice how these are unsaturated fats. Anyway, so water can diffuse across the cell, but it doesn't like to. It prefers to go through these aquaporins. And these aquaporins are just for water. So osmosis is the movement of water through the cell membrane, and it is a facilitated diffusion. So you might see diffusion of water, or you might see facilitated diffusion of water. Either way, it's referring to osmosis. So, more about osmosis. So the diffusion of water goes through a selectively permeable membrane from high to low. That is osmosis. So again, we're going from high to low. We're doing this without the use of energy, and we're going with the concentration gradient. So, we know water makes up a huge amount of your cells, so it's important that we talk about osmosis. Now, mentioning contractile vacuoles again, you can see in this picture here, they do look like suns. I showed you this in the last PowerPoint as well. But the contractile vacuoles are mainly for like protists or things that live in fresh water, and they help them contract, and so this will squeeze extra water out. Okay, So solute, solvent, solution. So a solution is going to be very simple. It's a solute and solvent mixed. Okay. So a solvent is going to be the substance the solute is dissolved in. So water is the universal solvent, and we mentioned that before in the last unit. Now the solute is what is dissolved. This is what gets dissolved. So you think of the solute as being salt or sugar or anything you would dissolve in water. Okay, so the solution is going to be your salt water. Okay, so thinking about Kool-Aid, your Kool-Aid powder would be the solute. The solvent would be the water. The Kool-Aid drink would be the solution. So again, water is a universal solvent, so it can dissolve lots and lots of things, and that's because of its polar nature. Now, tonicity is the concentration of water versus solutes in a solution. So osmotic pressure, that is the pressure that osmosis stops at, even if your concentrations are not equal on both sides of the membrane. So typically, osmosis would stop if both sides are equal on both sides of the membrane, like if you had equal amounts on both sides of the membrane. But osmotic pressure is when water flow stops and they are not equal. So, a couple things you need to remember about osmosis. Osmosis is only the movement of water. Okay, in order to understand the movement of water, you have to understand the solute concentrations. But again, you're still looking at the movement of water with the solute. It's a solution because water doesn't usually exist by itself. Now, if it's movement of solutes, that's probably diffusion or facilitated diffusion. Okay, but if it's movement of water, it's osmosis. The other thing is that it moves water molecules across the semipermeable cell membrane, and this is because the whole solution cannot pass through. So only the water is moving, not the solute. Okay. Again, if the solute is passing through, then it's going to move through regular diffusion or facilitated diffusion. We would not call it osmosis if the solute was moving with the water. So you have three types of osmotic solutions, and you do need to understand these three types. This is probably one of the more difficult sections from this PowerPoint, but it's not difficult. It's just one of the things you got to learn. But anyway, so there's three types of osmotic solutions. So the first type, and the easiest type for people to remember, is isotonic. So if you recall, the prefix iso means equal. Okay, so there's no net water movement with isotonic solution. And that's because, like if 5 goes in, then 5 come out. Okay, so the concentration, so that's the amount of dissolved substance, that's the solute, inside the cell is equal to that outside of the cell. Okay, so let me show you. Here's a picture of a beaker. Okay, so in this beaker you have a solution. Okay, oop, what I just did. All right, so that solution, let's say it's 10% salt, and then the other 90% will be water. You put a cell in that solution. Okay, the cell is also 10% salt and 90% water. Okay, so because of that, you have equal amounts on both inside and the outside of the cell. So if 5 go in, 5 will come out. And again, this is water movement. So because of that, the cell will stay the same size. Okay? So water's still going to randomly move, but there's no new movement of water. Now, plants are a little different, because then they may appear limp, wilted, or what we call flaccid, due to not enough water to totally fill the cell wall cavity in an isotonic solution. So that's kind of like an abnormal, compared to like animals, that would be weird. Okay, so here is some pictures of an isotonic solution. Okay, so one thing I wanted to point out to you here is you have arrows going in. Oops, sorry. You have arrows going in and out. Water is moving both ways. Okay, so equal amounts in and out. Notice arrows going in and out. Okay, and another thing, in and out. So it's indicating that equal amounts are going in and out. Now I want to point out to you that a lot of times we see these pictures of these red blood cells. Isotonic is really kind of what you would want, and notice it kind of looks like you have a thumbprint. Y'all see the kind of thumbprint, lifesaver type appearance that you see here. So if you see that kind of thumbprint looking red blood cell, then that's a normal red blood cell that is in an isotonic solution, and that's what you would want. Okay, so again, here's the exact picture I drew. So 10% salt versus 10% salt, water's going to move both directions. And now a hypotonic solution. So we're going to get more into the nitty gritty here. So the first thing I want to tell you is water follows the solute. Okay, so what that means is that water is going to travel to wherever the most solute is. If you have more solute outside of the cell, water's going to go out. If you have more solute inside of the cell, water's going to go in. Okay, and that's that whole traveling from high concentration to low concentration. Okay, so let me show you what I mean. So again, I have a picture of a cell in a solution. Okay, let's say that's still a 10% solution. Okay, so 10% salt. So 10% salt is going to be 90% water, right? Okay, so if that cell has 20% salt, where's your high amount of water? Your high amount of water is on the outside, so water goes from high to low amount because it would be 80 on the inside. So high to low, water movement, or you can think of it the opposite way. Water follows the solute. The most solute is 20% inside of the cell. So again, either way you look at it, water goes in. So water moves into the cell, therefore causing the cell to swell. So that swelling has to do with turgor pressure. So this is turgor pressure that exists in a plant cell. So the more water that gets in the cell, the more turgid it is, which gives it a more rigid feeling. And so it's a higher pressure. Now, this doesn't happen in plant cells, but in animal cells, this can actually cause the bursting of an animal cell, and that's called cytolysis. Lysis, if you recall, is like breaking open or splitting. So anyway, let's analyze this definition very closely. So you have the concentration, so the amount of dissolved substance, so that's your solute. So the definition, this is a solution, not the cell. It's talking about what solution you put the cell in. Okay, so what was the water like in that beaker? Okay, so it's talking about the amount of solute in water is outside of the cell is lower than inside. So it's always going to be referring to the concentration or the amount of solute outside of the cell. And it compares it to inside. So hypo, the prefix hypo means less than or under. And that's what's important for you to understand, that it's talking about the amount of solute outside the cell. So that outside of the cell, you're going to have less solute compared to water. So that's what this definition is saying. So because outside you have less solute, that means you have more, wherever there's less solute, there's more water, right? 90% would be more water compared to inside. So water follows the solute into the cell and swells the cell in a hypotonic. So hypotonic, you can think of hypo like a hippo, because hippos are fat. Okay, they are swollen. So if you look at this picture, you can see a red blood cell bursting. If you see that bursting, then you will know that this is hypotonic. The cell is filling with water. Notice all the arrows are going in because the cell is filling with water. The cell will swell. Okay, so again, this is a similar picture. Wow, I drew the exact same thing again. I did not mean to do that. So if you have 10% salt on the outside, then that means you have 90% water. Okay, and then you would have 80% water here, because it's either, whatever's left of it will be the water. So water is going to travel from the high amount to the low amount. Water will follow the solute. Wherever the most solute is, water will go in. So however you want to learn it. Again, this is water traveling. So water goes from high to low using no energy, or you can say that water is following wherever the most solute is, which would be inside. The diagram is showing the process of what? Hopefully you notice all the H2Os. So hopefully you know that that is osmosis when you see water. Okay, so that brings us to the last type of osmotic solution. That would be hypertonic. Okay, so hyper means more than or like over. Okay, so water is going to go out. This is the opposite. I told you earlier you could learn things opposite. So passive transport is the opposite of active. Hyper is the opposite of hypotonic. Okay, so water goes out. So again, this is the amount of solute is greater, because hyper means greater, in the water outside the cell compared to inside. So this causes the cell to shrink. So plasmolysis, this is the loss of water from a cell based on that drop in turgor pressure. So this is what causes a plant cell to wilt. It also causes shrinking in animal cells, which is called crenation. So anyway, here's your solution again. Let's do some different amounts here. How about we say, let's say we have 75% salt solution, and inside of the cell we have 1% salt. So it means inside of the cell you have 99% water, right? And then outside you have 25% water. So again, water's going to go from high amount to low amount. So from the high amount of water, 99% inside the cell, to 25% the low amount of water outside of the cell. Or you can recall that water follows the most solute. So the most solute is outside of the cell. So again, water goes out. So this will shrink the cell. Okay, so if you look here, hypertonic, we have a little hyper here, little Red Bull. Notice how these cells are like all jagged on the sides. That's because they're shrunken. Okay, so that's like a shrunken cell appearance that you would see for hyper. Notice all the arrows are going out because all the water is leaving the cell and it's shriveling up. Okay, so here's a picture again, a little bit different from what I drew. But again, if you have 20% salt, that means you have 80% water. If you have 10% salt, then you have 90% water. And water will travel from high to low amounts. And salt will go to wherever the water will follow the solute, wherever the most salt is. So water will leave the cell, shrinking the cell. So this would be a hypertonic solution because it's wilted, it lacks water. Okay, so again, here's a few more pictures. It's going both ways, so it's ISO. Notice the thumbprint. Okay, in this picture, you have all the particles are spaced out pretty equally. So in here, we only have the large dots in this picture. So hypotonic, water is going to go in. So you have all the little bitty dots are water. So you have your high amount of water on the outside and your low amount here. And you only have solute inside of the cell. That's the bigger, fatter circles. And then on this picture, notice up top that all the water is going in, so it looks swollen. Now in this picture up top, it's very shriveled up looking, and all the water is leaving the cell. And notice how many more solutes, the bigger circles, there are on the outside. So more solute on the outside, that means less on the inside. That means more water on the inside. So your high amount of water is going to travel to your low amount of water. What is the solution in this process called? Okay, so it's looking at higher concentration of substance outside of the cell, and then the substance will move in the cell. So that would be high to low. And so hopefully you knew that that would be diffusion. Okay, this is not isotonic because there's no net movement. We've not talked about active transport. It does not go high to low though. And we've not talked about mitosis. Okay, so this brings us to active transport. Like I said, it's the opposite of passive. So it requires energy, and energy is ATP. It goes from low to high amounts, okay, and it goes against. So active goes against the concentration gradient, and so that's referring to the low to high. So again, if you think you have 50 people in a room and it's very packed, it's going to be harder to go from the low amount of people in the hallway and get inside of that room that's already so packed full. It's going to be hard to go from low to high amount. Now, some of these active transports are going to involve proteins, whether they're a carrier protein or a protein pump. So again, active transport requires energy. Okay, so like the carrier proteins used in facilitated diffusion, you have these protein pumps. If you see pump, it will be active transport. Okay, and so it binds to that substance and helps pump them across the cell membrane using energy. And it's a pump, so it goes against concentration gradient. So it's going from low to high amount. So anyway, these are energy requiring because it's a lot harder to push them across the cell membrane. And so those pumps can even change the shape of the protein to allow certain things to stick to the receptors in the cell membrane. When a molecule binds to a channel protein in order to move across a selectively permeable membrane against a concentration gradient, the process is called what? Well, hopefully you remembered against is active transport. These are all types of passive transport, so typically, they would not be your answer. So this is active transport. Okay, a couple reasons I know these three pictures are active transport is, one, my arrow is going from low to high. Okay, 2, ATP is present. We have our low amount. So here's my low amount up here, and it's moving to my high amount down here. So that would show a active transport. And here we have ATP again. It actually doesn't show you an arrow indicating which direction the things are traveling, the little red things are traveling. But because ATP is present, energy is present, I know this is active transport. The process by which molecules move across a membrane from low to high concentration is what? So if it's going from low to high, then it's going to be active. All the others go from high to low. The channels in cell membranes that help substances move in and out of cells during active transport are made of. Okay, well, the only channels that are in the cell membrane are going to be protein channels. All right, so you have three types of active transport: endocytosis, exocytosis, and the sodium-potassium pump. So endocytosis is taking molecules in the cell. Okay, so this is taking large molecules in. Because they're so large, that takes energy. So this is moving things into the cell through a vesicle. That's called endocytosis. So this is going to be bigger molecules, groups of molecules, or possibly even a whole cell. So what happens is part of the membrane breaks off and kind of circles around maybe that molecule, that group of molecules. It circles around it and makes a little vesicle. And then it goes to wherever it needs to go in the cell. So this is just showing a general picture of endocytosis. You have all your molecules coming up here, and then part of your cell membrane, notice, is folding around it, forming a vesicle. So that's endocytosis. Okay, so you have three main types of endocytosis. Again, we've mentioned this before, some of these. So cell drinking is penocytosis. So that's going to be fluids. The cell taking in, because it's endocytosis, fluids. Phagocytosis is cell eating. Okay, so the cell taking in large particles or whole cells. So it's cell eating with phagocytosis. And again, this is taking in things because it's endocytosis. So an example is an amoeba eating its food through phagocytosis, or a white blood cell eating a bacteria through phagocytosis. And the last one is receptor-mediated endocytosis. So this is where particles bind to the proteins on the surface of the cell, allowing you to bring in bulk amounts of things, so large amounts of things. So it's a spot where it binds on the surface of the cell. So anyway, the particles are drawn into these things that are called coated pits, and it forms like a cell surface. And you'll see ligands in that process. Anyway, the pits collapse into a vesicle that's been coated, and eventually it becomes a vacuole called an endosome. So remember, vacuoles store things. So here's a picture of each one of these. So you have your phagosome here, your phagocytosis, where part of the cell membrane pinched around it. Then you have pinocytosis, where it's just cell drinking. So it's taking in a large amount of fluids. And then you have your receptor-mediated endocytosis. And notice that this receptor fits this certain thing here, and it goes into this coated pit to make a coated vesicle. And again, this is endocytosis here. Pinocytosis, the cell taking in a large amount of fluids, and then it buds around it to form a vesicle. Phagocytosis, it eats the food goes in, and part of the cell membrane goes around it to create a new vesicle. Now, so the next part is exocytosis. Okay, so exo is exiting. So this is the opposite of endocytosis. So this is where things are moved out of the cell. So a vesicle will approach the cell membrane and eject waste. And notice the walls of the vesicle will absorb back into the cell membrane. So if you're thinking with endocytosis, maybe you would lose some of the cell membrane, well, you would gain it back when the wastes are produced. So this is how vesicles travel to the cell membrane, and then waste are ejected from the cell, or even hormones if it's a hormone-producing cell. Okay, so this again shows exocytosis. So we have, actually it shows endo and exocytosis. So endocytosis, you take the food in, and you got your food vacuole, then it combines with your lysosome, they digest it, and then the waste products are then released through exocytosis. Okay, again, exocytosis, you are releasing waste from the cell, and this is, again, exocytosis, releasing waste from the cell. So the last example of active transport is the sodium-potassium pump, which is critical for survival. It is a pump, so it is going to use carrier proteins, and it moves 3 sodium out, so sodium is Na, and 2 potassium in. This is why you use or need more sodium than potassium. So what happens, these are the steps, the numbered steps here. So first 3 sodium are going to bind to a transport protein on the inside of the cell. And then ATP is going to change the shape of that protein to eject the sodium out of the cell. Then 2 potassium from outside of the cell are going to bind, and it changes its shape and releases the potassium inside of the cell. Okay, so the sodium-potassium pump, it keeps all your nerves sending messages, like your brain sending your heart the message to breathe, and it also allows your muscles to continually contract. And we do have some muscles that are necessary for survival, like the heart muscle, and some muscles that are associated with breathing, like the diaphragm. So, one thing you might not have known is that 1/3 of your energy at rest is used to power the sodium-potassium pump. So, lots of energy in every cell in your body is used for the sodium-potassium pump. It can transport 450 sodiums and 300 potassiums in 1 second in one cell. So here's a picture of the sodium-potassium pump. And I know it's a sodium-potassium pump because I can see, I can see the NAs, the sodiums here, the NAs, the sodiums, and I can also see the potassiums. Okay, and I can also see energy, and it's moving through that protein. Which of the following substances can most easily travel into and out of the cell? So I hope that you're thinking about the thing that makes up the bulk of cells, and that would be water. All right, so that concludes this lesson. So this was a little bit shorter than the last one, and I hope that you found this beneficial for you. Make sure you are doing the practice problems, because this is one of those things where you have to apply the knowledge that we looked at.